All right, so for today, we are still talking about DNA, and let me just zoom this screen out. And we're going to be doing some demonstrations and doing a lab today with DNA, but first we need to make sure that you understand what's going on. Just to go over, you do have homework today. This actually should say complete your DNA worksheet, so I'm going to change that there. But your homework is to complete your strawberry DNA extraction. You will have to complete that for homework, and it's due in class tomorrow. All right, so let's then take a look at the picture that you needed to draw. You look at this side of the screen. With color pencils, you should have drawn this picture, and then you had to label some things. So this portion here, what would this be considered? What is this circling? What is it? Why is this a nucleotide? It includes the base, which is where? What color? Red or orange? The ribose sugar, which was what color? Black. And then this is this should have a white part here, but that would be the phosphate. So this is the nucleotide. If you did not label that nucleotide, you want to go ahead and make sure that you do that. This portion here, what did you label this as? You said phosphate group. Anyone have anything else? This is your sugar phosphate backbone, because if you notice, it's outlining the sugar and the phosphates. And when it says backbone, remember, these are the rails of your DNA. So for instance, remember we made the DNA yesterday. These would be the rails of the DNA. What is the shape of DNA? Is it shaped like this? What's the shape of it? A double helix, like a twisting staircase. So we were to put them all together, DNA is shaped like so. Understood? All right, what about this here? The yellow-blue pairing. That will be a base pair. Good job. Tell me something about base pairs. Justin. They only match specific bases. Now, it isn't ironic, or not ironic, but very unique that there's only 4 bases in DNA. That's every DNA. Your DNA, a strawberry DNA, a banana DNA, a chimpanzee DNA. There's only 4 bases. But the way that they are organized is what makes you you. I only have 4 bases. Messiah only has 4 bases, but we look nothing alike. We act nothing alike. We have different bases. Same thing with you, as well as your counterpart, as well as somebody on the streets that you don't even know. All right, what about here? These would be just considered they're pointing to them separately, so they're considered your bases. Here you have a base pair because they're looking at them together, and here they're pointing to them separately. And then this black thing is the black thing, everyone? The black thing. Say it louder. This would be your sugar. All right, so we're going to go ahead and move on. In your trays, if you go ahead and put everything away, because we're going to go ahead and get started on our lab. I'm going to be going over some materials with you so that we can get started. You can even put your homework sheet away. Your area should be totally clear. If at your station, if you can take out, pull a paper towel. You'll be fine right now, Darren. Alrighty. So I'm going to be going over your materials. On the right-hand side of the screen, you will notice there's a video there. I did go ahead and do the lab so that it's already there. We're going to do it together with you. I'm going to play the video and go along and speak with you. The video does not have any, hit the bottom light for me. The video does not have any sound, and disregard how I look. It was last night, so yeah, disregard the crazy hair. Alrighty. So, in your bags, you do have a Ziploc bag. Identify the Ziploc bag. Everyone in your labs, identify the Ziploc bag. Everybody. Well, not everybody. I apologize. You're doing each side of the table is your group. So it's going to be. Let me stop. I apologize. Let me backtrack. One bag for the entire table, but then after that, you're going to be working for each group. So just take out one bag for the entire table. Now, instead of playing the video, actually, well, I'll play it in a moment, but what you're going to do is, first of all, we are going to be extracting DNA from a strawberry. Now, why would we say that a strawberry has DNA? Why is it alive? What do you mean? It's a plant. So plants obviously have DNA. Does all plants have DNA? What else has DNA? Animals, people, we're animals. Bacteria has DNA. And a strawberry has 2 sets of 7 chromosomes. So how many? 14. Where did it get it from? Mom and daddy? Where? The seeds. Where do we get ours from? Mom and dad. Okay, so we have a bag, a plastic bag. What we need to do first is if you go ahead and take the strawberry leaf and pull it off. Just pull it right off. Take the strawberry, go ahead and put it in the bag. Push the air out and seal the bag. And then what you're going to do is one person, that same person or somebody else, go ahead and squish the strawberry in the bag. Make sure the air is out of the bag. And just like the video, squish it all up. Squish the strawberry in there, smash it so that it's evenly smushed, kind of like you want it to kind of look like a slushy, a strawberry slushy. Make sure there's no lumps. You want to squeeze all your little lumps out. Make sure there's no lumps in there. You need to smash it some more, Shania. Smash it with your hand like this if you need to. Smash it all up. Pass it around. Let everyone get a chance at smashing up. Just make sure none of it seeps out. But you really want to smash it up like I'm doing there. And I'm smashing up all of the little knots, the little... Now, what do you think the reason why we're smashing it up? No, not to see what's in it. Where is DNA located? Inside of what? Inside the cell, but where inside the cell? Inside the nucleus. What protects that cell on the outside? Cell membrane. This is a plant, so it would be the cell wall. So we need to go ahead and mash up that cell wall, break that cell wall so that we can actually get inside of that cell so we can actually access that nucleus. Does everyone agree that your strawberry is all smashed up? Okay. Then what you're going to do, in your packet or in your box, you have a liquid that's titled cell lysis solution. You want to take this solution out, and we're going to do this the one time for the bag. I'm trying to make sure this is focused. Lysis means to cut up, to split up, so this is going to help the cell membrane split up even more. In your graduated cylinder, the hard plastic one, if you take a look at the video, this graduated cylinder, not the little one, everyone has a longer hard plastic one that looks like this. You're going to go ahead and take your cell lysis solution and you're going to squirt it into there up to 10 milliliters. That's the very top notch. It's probably going to bubble up like dishwashing liquid because it is a soapy solution. Then go ahead and open up your squished up strawberry bag and pour that in there. Before you seal it up, you do want to squeeze out all of the air bubbles. Make sure when you close it back up, there's no air bubbles, so you have to kind of push your bag up from the bottom without letting any of the solution seep out. But go ahead and pour that right in there. Before you seal it, you got to get the air bubbles out before you seal it. And then we're just going to let that sit for a minute. So I'm taking all the air bubbles out. Now let's talk about this for a moment. What do you think that the DNA of the strawberry is going to look like? Well, first of all, what does extraction mean? To take out. So that means we're going to be doing what to the strawberry? Taking out the DNA. What do you think it's going to look like? It's maybe stringy. Okay. Anybody have anything else? Don't do that. Just let it sit. What else do you think it's going to look like? These are guests, so please talk to me. Slimy, watery. Do you think you're going to be able to see it? You don't think you're going to be able to see it?
I do.
You think you are going to be able to see it? Do you think it's going to come out looking like this?
No.
No? Is it going to be as sophisticated as our DNA?
No.
Not necessarily, but remember, when you look at a DNA molecule and it shows you a sample that looks like this is magnified by a lot. Remember, our cells, we can't even see with the naked eye, so quite frankly, we can't see our own DNA, but we are going to go ahead and pull this DNA out of here. We only have a couple more seconds before our time is up, and the time is now up. So what you're going to do now is you are going to take your piece of cheesecloth. The cheesecloth is actually the little piece of fabric that looks like this. Only pick up one. If you take a look in the video, you are going to go ahead and put it in the funnel. But I want you to be very careful because if you notice, cheesecloth is very thin. If you take it and lay it over the top of the funnel like so, and each group is going to do this. So each side of the table, we need to now do this. You're sharing the bag, but one side is going to pour some, and then the other side is going to pour some. So you're going to take your funnel that looks like this, and then you're also going to take your smaller graduated cylinder, the one that looks like this. There is a base. You're going to sit it in the base. Sit your funnel inside of there. The one that you're going to see in the video, I didn't use this type of graduated cylinder that we're using in here. I actually used this one, but that's perfectly fine. In a moment, you're going to go ahead and take turns. You're going to pour, you open your bag and you're going to pour some onto the cloth, but you have to make very sure that none of it gets on the side so that you don't want big pieces in the funnel. You only want the liquids. Now, this cheesecloth is acting like a coffee filter. When you make coffee, you don't want to drink coffee with a bunch of grains and beans in it, correct? You actually want it to just be the liquid. So we are filtering it out using the cheesecloth just to have the liquid. So please make sure when you are having this, one person can actually even hold this over until you've poured enough where it's kind of thick and it's going to sink down, but you don't want it to get in the sides. So if you just take a look at the video for just a moment, you notice that my cheese filter is in there. And then I'm going to open up the bag. Again, I'm making sure that I'm not going to be getting anything on the side. So one side of the group can go ahead and open up the bag. Don't pour it all, you're just pouring some. You want to at least get maybe, I'm sorry, I was just thinking of something. You want to at least get maybe 5 to close to 10, but you're just going to pour it in there because we're going to need to let it sit for about 5 minutes. So pour some to where it's in there, then switch with the other group and let them pour some. Be poured slowly so it's not getting, you can let the filter sink down. Give the other group the bag. No, no, no. You guys are doing it a second time, unless you all just want to do it once. What I'm saying is you're going to have 2 graduated cylinders with 2 funnels and 2 cheese filters. But since you've already just done that, we'll just do it once there. Like they have 2, each table has 2, but we'll just leave it like that. Go ahead and pour some more in there since you guys are only doing it one time. Do it slow, Mike. And again, we're going to let this sit for 5 minutes. And then give it to them so they can pour a little bit more on. So, and I'm just letting mine sit there and go until the timer's up. So we'll just wait a moment while the time is up. I want to ask you a couple more questions. Let's do a little bit of review. Tell me the 3 parts of a nucleotide now that you're not looking at your notes. Phosphate, sugar, base. Tell me something about the phosphate, like where it is. At the bottom, what do you mean? Well, let's think about it. A DNA, we can compare it to what? A stairwell or a ladder. So if we're comparing it to a stairwell or a ladder, where would the phosphate be? The rail. All right, then you said there's a sugar. What would the sugar be? It would also be part of the rail. It connects the rail. What would the bases be? Those would be the stairs or the bars that you're climbing up on a ladder. Now, there are how many bases? 4 bases. Raise your hand and tell me what those 4 bases are, Justin. Adenine, cytosine, thymine, and guanine. Now, the thing that makes DNA so unique is something to do with the bases. Really has nothing to do with the sugar and the phosphate. It has to do with the bases. Why do I say that? Why do I say it all has to do with the bases? Only Emmanuel and Justin were here yesterday. Nakaya. Okay, how they're set up. So, for instance, one of my sides of my rails could be adenine, guanine, cytosine, 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 guanine, guanine, thymine, adenine, adenine, correct? Okay, yours could be something totally different. The pairing could be totally different. How many do we have? How many chromosomes do we have? We have 23, two sets of 23, which means we have 46 chromosomes. We got 23 from our mom and 23 from our dad. Let's go back to these base pairs. Just raise your hand. Don't yell out you. I will eventually see you. I'm sorry? Twins do have, identical twins do have the same DNA. That's what they look exactly alike. They have the same DNA. All right, so the pairing or how the DNA, how the bases are actually labeled, what else makes them unique is they only match with specific other bases, correct? So adenine only goes with thymine, cytosine only goes with guanine only goes with thymine only goes with adenine. Now, once these things are paired up together and they make our DNA is all wounded up and all put together, and it tells us our what? Say it louder. It tells us our genes, which identifies our traits. What's a trait? The things that make you. How you act, how you talk, how you look, how you walk, what your skin is like, what your hair is like, how far apart your eyes are, how big or small your ears are, things of that nature. Those are your traits. Now, those are all wrapped up into these little X's that are within the part of a cell. What are those X's called? Chromosomes. And again, you have how many? 46 chromosomes. Now, with that being said, they're all wrapped up and they're all put inside the middle of a cell, and that middle of the cell is located where? In the nucleus. So is it safe to say the chromosomes is smaller than a cell? Yes, the chromosomes are smaller than a cell. All right, it's time to go ahead and do something else. Our time is up. So what we're going to do right now, you have a test tube. Each group is going to go ahead and take out a test tube. And then there is something in your container called a DNA, maybe a DNA extractor. What is it? DNA precipitation. It's cold because it has to be cold. Precipitation, what's precipitation? Rain. Not just rain. Rain, sleet, star, hail. That's an example of precipitation, but precipitation is just creating of something that wasn't there. For instance, you have a liquid that formed from a gas, or you have a solid that forms from two liquids. So this is going to be able to form or capture that DNA that's there, because right now you just have a bunch of liquid, so it's going to be able to pull and link that DNA together. So you have your DNA precipitation, and what you're going to do in your test tube, which is the glass, be very careful. You're going to go ahead and put about halfway, you're going to go ahead and pour some DNA precipitation in there. All right, are you done? Just halfway. That's good. That's good. That's good. Not too much. Just halfway. Notice, some of you have already identified and noticed that I put these trays here. Once you take everything out of there, you can flip the trays upside down and use it as a test tube holder. But right now, you're not going to put it in there, because what you're going to need to do, the two people in your group are going to need to work together. If you take a look at the video, it's just me, but what I'm doing, you're going to hold this. Don't do it yet, but you're going to hold your test tube at a 45-degree angle. One person is going to do that. Another person is going to go ahead and take their cheesecloth funnel off and set that on the napkin. Take the whole funnel. Take the whole funnel out. Yep, take the whole funnel. Take that whole thing out. Yep, Tim. Set it on the napkin. Don't take the cheesecloth out. Take the whole funnel out so that no pieces get in there. Messiah, go ahead and take the whole funnel out. And it's okay if it gets messed on the lab station. One partner is going to go ahead and hold the test tube at an angle. Another partner is going to take your dropper. And that partner holding the dropper, as you can see, if you look in the video, I've pulled up some of the strawberry mixture. You're going to pull some of that up in there. Now, watch the video for just a second, because what I'm doing is I'm going to put some of that strawberry solution in my test tube, but very slowly. So when you squeeze on the dropper, you're going to squeeze it very slowly so that the strawberry solution is sliding down the side into the DNA precipitate. Okay? And you're going to do that until your test tube is to about here with liquid. Just a little bit more, like 3/4 of the way. So go ahead and do that now. Yes. Yep, you take it out of you're going to be taking the strawberry solution from your graduated cylinder where you filtered it. So that should be about maybe 4 of the droppers of the strawberry solution. Once we're done with that, we're going to let that sit for 3 minutes. So once you're done putting in there, go ahead and sit it in the test tube holder. And then we're going to let that sit for 3 minutes. So let's let everyone get that situated. It's fine as long as you got some in there. Then go ahead, everyone, sit it in the test tube container. If you're putting one more dropper in there, let this be your last one. I'm going to go ahead and put the timer on for the 3 minutes. While that's doing that, let's talk some more. So we talked about how our DNA is raveled up into your genes, raveled up into making these chromosomes, into your nucleus. Let me ask you, what are some the fact that we're pulling DNA out of the strawberry, what does this mean for all of the fruit that we eat? It all contains DNA. What about your potato chips? Why? Because it came from potatoes. What about your hot fries, Jamaica's sister's favorite food? All of you like those hot cheeses. What about those? Those have DNA? Did they actually have some DNA in it? Why? Some of them may have come from potatoes, but they also came from what? A plant, wheat. And the wheat comes from a plant, and a plant is. What about Stefan's Frescom from Steak and Shake? I haven't had one. Yes, why? It's beef. So it's from the. Now, here's the thing. Interesting fact. The interesting fact about beef, as opposed to vegetables and fruits, the DNA in beef and vegetables and fruits, that DNA actually starts to link and coil with our DNA. So that's the pretty weird thing, but we need to get going. So something should have happened already. Go ahead and lift up your tube, and you will notice a white precipitate right in between the clear liquid and the red liquid. It does. You have a tube like this in your container. Go ahead and stick that in there and try to pull it out so that you can look at it. What does it look like? This yellow thing, yeah. The pointy side. Or either side actually will work. What does it look like? It looks like snot. It's cloudy looking. Like a spider web. All right, so you're going to use this information to your knowledge and your experience from this lab to complete your homework today.